Hi, everybody. Welcome to another Chats with Children. I hope you're safe and well. Today, I'm delighted to be joined by Ken Cook, who is heading up the European Pharma Bio Applications Support team at Thermo Fisher Scientific. And Ken and I have not spoken for over 18 months. It seems like 2 years, a long time actually, but I'm glad to say Ken is looking well. So Ken, how are you, my friend?
I'm fine. It's looking good. The weather's starting to perk up, so it's everything to look forward to.
Yeah, well, it is sunny down south, as you'd say, because you're up in the far north in Northumberland. So I'm glad to see it's sunny where you are as well, Ken.
Yeah, it's usually good. I'm on the East Coast. The East Coast doesn't get the rain.
Fantastic. Now, Ken, the reason we're actually talking is because you are part of a new hybrid roadshow that Thermo are running throughout the year, all around oligonucleotides, correct?
That's correct.
Fantastic. And today we're actually going to be talking about why has oligonucleotides analysis become so important? So before we talk about that, maybe for people not familiar about oligonucleotides, you can talk about why they are important in the first place.
Yeah, I think when you look at oligonucleotides, they've always been around, and they've been used in research labs. However, delivery into the human body as a medicine has always been a bit of a stumbling block to get over. But everyone knows about the COVID messenger RNA vaccine unless you were living in a cave. But that messenger RNA vaccine proved that this could be safely achieved just by encapsulating the oligonucleotide in a lipid nanoparticle. Now, that lipid nanoparticle helps aid entry into the cell, which has also got a lipid outer layer, so it merges with the cell and gets the messenger RNA inside the cell, but it also protects the drug from inherent nucleases that are in the body. Otherwise, as soon as you inject an RNA molecule into the bloodstream, it will just get eaten away. So now you've got a way of actually getting the messenger RNA into the cell. And because of the COVID vaccine, we know that messenger RNA codes for a protein, and the body produced that protein, pushed it out, and then you've got the immune response. So we can now get messenger RNA inside a cell and produce a protein. Now, this opens a huge door for new medicines, because it's not just the fast production of vaccines, which is great in its own, but you've got many genetic states where the body fails to produce a protein, or it's producing a protein that's bad that it doesn't need. Now we've got a way of actually preventing that and curing some diseases that just did not have a cure before. And there's a lot of genetic diseases that did not have a cure.
Right. So you sort of answered my next question, I think. But so why has oligos, I'm going to cut it down, so I always struggle to say oligonucleotides, analysis become so important?
Well, imagine you've got a shop and suddenly a brilliant new product comes up. All the shops want that new product and make different versions. It's like having a sweet shop and someone says, "I've just invented sugar." But you've got a lot of store open, you've got a lot of disease states now that are new targets, but in the biopharma companies, every single one of them is doing this now. They're all stepping up to the new challenges, and I mean all of them. And they're all working on small interfering RNA to combat bad proteins, stop the production of a bad protein, and they're all working on messenger RNA for vaccines or to produce a protein that a body needs that it doesn't normally have. But at the end of the day, this is a new market, but they've all got to be characterized to make sure they're safe. And that's why a lot of people are now looking at oligonucleotide analysis. It's a new modality, and these people have to be able to make sure that new drug is safe when you inject it into the body. But you're looking at analysis labs that have never really been used to using oligonucleotides. Now they've got to come up and develop methods or use methods they've never used before. And to be good on this one, the FDA fast-tracked the COVID vaccine. And they are fast-tracking some of these diseases because these new drugs, because they're hitting diseases where there's no new cure, no cure at all. So if you've got a patient and these patients are dying, you need that cure. So FDA is coming up and stepping up to this challenge as well. So it's all looking quite good for some of these new diseases that are being targeted, and patients that really did not have any hope before have now got hope.
Right, but you talked about obviously the FDA analysis, but what are some of the common ways people have historically done the analysis, and what are some of the challenges that people face when they're looking at these applications?
Well, you've got the small molecule drugs, say aspirin. It's C18 column, HPLC. Manufacturers know exactly how to do that. Now we're faced with a synthetic molecule that's highly charged. It's much bigger. It's got many more impurities. Aspirin, 2 or 3 impurities. These have got quite a few possible impurities. They all have to be characterized, and they need new methods for this. And it's not easy to run these methods. An oligonucleotide's got a long phosphate backbone. It's probably the best known natural chelator in the world. Steel is difficult, metal ions are difficult. So there's a lot of problems when you come to actually analyzing an oligonucleotide compared to the small drugs these labs are used to seeing. So there's a lot of new developments to help this, but in essence, there's a lot of people new to the technique, new to the analysis, that need trained up on how to do this.
Right, and you said there are lots of new methods. What are some of the new methods then that people are using to help analyze oligonucleotides?
Yeah, well, if you've got a protein drug, say a monoclonal antibody, people have digested, sequence it, yes, it's the right protein. That analysis is analogous to a messenger RNA, a big oligonucleotide, but how do you digest it? How do you sequence it? And these are new methods that we've done. It's the only method that's only been available in the last year, but we can now sequence messenger RNA similar to the way we sequence a protein. And then the small molecule drugs, same thing. It's difficult to separate some of these small molecule drugs because they're very highly charged. And there's new columns, new techniques, new mass spectrometry methods for this, but also the software. The software that's used in the past for proteins, for small molecules, that needs now to look at something completely different. You've got an oligonucleotide. You can sequence a protein with protein sequencing software. Oligonucleotide sequencing software has never been available before. And again, that's available now as well.
Right. Well, as I said when we started this conversation, we're doing this obviously because you are part of a new roadshow that Thermo have launched and are launching with the first event in May in Hampstead, down south in the UK, called European Oligos on the Road and Beyond. So let's talk a little bit about that. So what are the sort of things you'll be covering in that, and who are the sort of people who will be talking and presenting as part of that roadshow?
Well, we've got customers, and they'll be sharing their experiences. They'll also be sharing the methods that they've developed to actually help analyze the oligonucleotides. We'll also be looking at doing a demonstration of the new software techniques and giving tips and tricks on how to deal with oligonucleotides. It's completely different to small molecules. The chromatography conditions are different, the columns are different, and again, when you actually go to use them, you can come into a lot of pitfalls that can be avoided if you actually come along to the seminar, and we'll go through these pitfalls, how to run these systems, and you'll get the experience of some very experienced customers who've been running oligonucleotides for quite some time.
Brilliant, Ken. Well, look, thank you for that, and thank you for giving me a quick overview of what's been happening around oligos analysis over the last year or so. So there you go, viewers. If you would like to learn more about this, then as Ken says, Thermo Fisher Scientific are running the European Oligos on the Road and Beyond series, kicking off on the 11th of May. It's a hybrid event, as Ken pointed out to me before we started this chat, which means there's a live audience at the venue, but also there is also a virtual audience as well, which you can tune into, and that will be running on 11th of May, 10 a.m. to 4 p.m. GMT. And some of the speakers, actually, I've got them in front of me, is Professor Mark Dickman, who's from the University of Sheffield, and David Schmidl, hopefully I pronounced that correctly, of the University of Cambridge, and Ken himself, we'll be doing a presentation of the Thermo's Biopharma Finder software. So check that out. The link will be above the video. But if you've got any questions based on what Ken was talking about oligonucleotide analysis in general, or anything specific, then feel free to put them in the comments below the video, because I'm sure Ken will be only too delighted to answer any questions you've got as well. Well, Ken, genuinely, it's lovely to see you again after such a long time. You are looking well, my friend. I hope you have a great show, and I hope the demo goes really well as well. And this is, I know, just the first of a number of different events. So again, check out the Thermo Fisher LinkedIn page and website to find out more details about future events as well. So Ken, thank you very much.
There will be a round table at the end as well, so we're inviting people to come up with their questions, and we'll be discussing it at the end.
Well, then in that case, if anybody has any questions for the roadshow as well, then please put them in the comments below, because I'm sure Ken will only be too happy, if I get my dentures in the right way, to have a look at those questions and add them to the roundtable discussion, the Q&A they're going to have at the end. So Ken, once again, I hope the sun continues to shine in the northeast. Viewers, I hope the sun is shining wherever you are today and for the rest of the week and for the year, all being well. And until next time, as always, stay well and stay safe. Bye-bye.